This is part two of the lecture on food systems and associated antimicrobial resistance.
In contrast to using antibiotics to treat an infection in an animal, widespread use of antibiotics in food animals became a problem after it was discovered that using antibiotics on animals that weren't sick could increase large scale production efficiency by increasing animal growth and could be used as a substitute for labor previously devoted to the care of individual animals. In addition to speeding up growth, giving food animals subtherapeutic doses of antimicrobials has been used to prevent disease in animals raised in overcrowded, unsanitary conditions. Non-therapeutic use of antimicrobials contributes to the development of antibiotic-resistant microorganisms, which pose risks to both human and animal health. In 1951, the FDA approved the use of antibiotics in livestock feed to promote growth. This practice became widespread, primarily driven by economic incentives. In 1961, the global average per capita meat consumption was 24 kilograms. It increased to 43 kilograms in 2014. This rising demand for meat meant more pressure to produce more and bigger feed animals at increasing speed. Recent projections predict that growing meat consumption in middle and low-income countries will lead to a 69% increase in global agricultural antibiotic use by the year 2030. The scale of the problem of antimicrobial overuse in food animals is difficult to quantify because most countries don't release data on agricultural antibiotic use publicly, and about 40% of countries don't even release this data to the World Organization for Animal Health. Antibiotic use in food animals continues to grow worldwide, even after being established as a driver of antimicrobial resistance and despite concerns about the public health consequences. This graph was produced by cross-referencing antibiotic sales from 42 countries for which data was reported, data on global food animal populations, data from individual governments, farm surveys, and scientific articles on veterinary use of antibiotics to estimate antibiotic usage in 229 countries. Note that the 42 countries that reported data were all high income, so antibiotic use in low-income countries may have been underestimated in this analysis. China currently uses more antibiotics in farming than any other country, but Pakistan is expected to experience the most growth in use from 2020 to 2030. The researchers also estimated that antibiotic use will grow the fastest in Africa, rising by 25% between 2020 and 2030, owing to the increased demand for meat products. These graphs from that same study show veterinary antimicrobial consumption in 2020 in the white bars and projected consumption for the year 2030 in the colorful bars. On the left is antimicrobial use by country, showing the top 10 countries. In the middle is use by antimicrobial drug class, and on the right is use by continent. Tetracycline stands out as the most used antibiotic in veterinary animals, and China stands out as the nation responsible for the most veterinary antimicrobial use. Recent regulatory changes in many regions have restricted or banned antimicrobial growth promotants, yet the practice poses an ongoing biosecurity risk. Global use of antimicrobials in livestock is in the tens of thousands of tons annually, but it's difficult to accurately quantify global use. Reporting has improved in recent years, and some high-income countries are showing sustained declines in use following implementation of antibiotic stewardship practices. But antimicrobial overuse in low and middle-income countries continues to increase in response to increasing demand for animal protein. Governments have struggled to rein in inappropriate use of antibiotics in food animals because regulation is viewed as counter to profits. Many pharmaceutical companies have catered to the economic incentives of food producers by labeling antibiotics as intended to prevent disease after sale of the same drugs, if intended to promote growth, was banned. Intensive livestock practices consume 4 times more antibiotics compared to livestock raised outdoors. While it's possible for antimicrobial resistance to develop at lower levels, 50 milligrams per population corrected unit has been proposed as a threshold below which development of resistance is less likely. Many countries around the world continue to use antimicrobials in animals above this threshold. Antibiotics were one of the most important advances in human medicine. Previously, common infections and surgical procedures were often fatal. Antibiotics add about 20 years to the human life expectancy. Soon after discovering penicillin, Fleming realized that antibiotic resistance could develop if too little penicillin was used or if it wasn't used for long enough. He cautioned the public and medical community not to use penicillin unless there was a properly diagnosed condition, and that it should only be used in sufficient quantities for sufficient duration to treat the infection. Otherwise, bacterial resistance to the antibiotic would develop. Unfortunately, we did not heed Fleming's warning. In 2015, about 80% of all antibiotics sold in the US were used in food producing animals. About 70% of those were medically important antibiotics. About 75% of antibiotics an animal consumes are excreted from the body in their feces and urine, which contaminates the surrounding environment, including water and manure that's later going to be used as fertilizer for food crops. The misuse of antibiotics in food producing animals has led to diverse antibiotic resistant bacteria and antibiotic resistance genes, which can be transferred in animals and humans. There's increasing evidence that antibiotic resistance in humans is mainly related to the wide application of non-therapeutic antibiotics in animals. As factory farms and confined animal feeding operations that rely on non-therapeutic use of antimicrobials expanded globally, so too have drug-resistant bacteria. MRSA, methicillin-resistant Staphylococcus aureus, and VRE, vancomycin-resistant Enterococcus, are likely two examples you're familiar with. Some of the strategies bacteria use to fight antibiotics include enzymatic degradation of the drugs, alteration of the bacterial proteins that the antimicrobial drugs target, and changes in membrane permeability to antibiotics. Antibiotic resistance can either be plasmid mediated or maintained on the bacterial chromosome. I'm going to go over an example of enzymatic degradation of an antibacterial drug, but we have to first talk about how antibiotics work when the bacteria don't know how to fight them. Bacteria primarily reproduce asexually through binary fission, where a single cell replicates its DNA, then splits into two identical daughter cells. Other less common methods include budding, spore formation, and conjugation. In binary fission, the bacterium copies its circular DNA, the chromosome. It elongates and pulls the two copies of its DNA apart, then it pinches off the cytoplasm and builds more cell wall to divide into two identical daughter cells. Antibiotics work by targeting prokaryotic cell components that humans don't have. Bacteria have a cell wall made of peptidoglycan, but eukaryotic animal cells don't have that, and plants, fungi, and algae that have cell walls build it from different components. In the presence of penicillin, when the bacteria tries to replicate itself, because part of the penicillin molecule looks like the building block of the bacterial cell wall, the bacteria try to use penicillin to build its wall. But it doesn't link together properly, so the cell wall produced won't be intact, so the bacteria's intracellular contents will leak everywhere and it'll die. E. coli is an example of a bacteria that frequently has beta-lactamases, which break apart the ring in the penicillin structure that normally gets included in the cell wall of bacteria when they try to replicate. When penicillin's structure is broken by beta-lactamase, it no longer harms bacteria. When bacteria have the instructions for making beta-lactamase in their chromosome, it can be induced through exposure to beta-lactam drugs like penicillin. Resistance to methicillin, like with MRSA, occurs through an alteration of the antibiotic target protein, in this case penicillin binding protein 2. The probability of bacteria developing resistance increases with increased exposure of bacteria to that antibiotic. This is true both in humans and other animals. The more we use antibiotics, the more pressure is put on bacteria to mutate or find other ways to survive in the antibiotic's presence. In addition to subtherapeutic doses of antibiotics, another way that resistance can develop is if antibiotics aren't used for long enough duration when treating an infection. For example, let's say an antibiotic is prescribed for 5 days twice a day. On the first day, the person takes the antibiotic as prescribed, and many of the bacteria are killed. On the second day, the person takes the antibiotic again, and even more bacteria causing the infection are killed. Some of the bacteria begin expressing a protein that their chromosomes already contained in response to the stress caused by exposure to doxycycline. Then the person thinks they feel better, so they decide not to complete the rest of the treatment. When the first dose is missed, the bacteria are able to reproduce again. More bacteria have turned on this gene that encodes the trait that confers resistance to doxycycline, so they pass it on to their offspring. And bacteria with the trait expressed are more likely to survive and reproduce as remaining doxycycline is cleared from the body because they have a survival advantage over bacteria not expressing this trait. The surviving bacteria that weren't killed by the antibiotic are selected for, and they're able to reproduce more freely because they don't have the competition of all their buddies that got killed by the antibiotic. So then you get a population that's all copies of that bacteria that were resistant to the antibiotic. So if the same antibiotic is used again, it'll have little or no effect. This example of inadequate treatment duration demonstrates selective pressure. When an antibiotic is used, bacteria susceptible to the antibiotic are killed, while those with resistance genes survive and multiply, effectively selecting for the resistant population. This is not a complete list of the mechanisms by which antimicrobial resistance develops, but I wanted to provide a couple examples. Another way antimicrobial resistance develops is through horizontal gene transfer when microbes share resistance plasmids. Once a single bacteria mutates to become resistant to antibiotics, it can transfer that resistance to other bacteria around it through conjugation, which involves direct physical contact. This cartoon depicts how a plasmid, a circular piece of DNA, is shared between bacterial cells. Antibiotic resistance can be spread on plasmids even when no antibiotics are present. Because remote regions of the Arctic provide an environment with little human contamination, researchers studied plasmids and assessed for the presence of antimicrobial resistance genes near Svalbard, Norway. They concluded that glacial ice may be a repository for pathogenic microorganisms because virulence genes and biocide resistance genes were dominant in their samples. Another interesting finding from the research was a strong correlation between antimicrobial resistance genes and metal resistance genes in the fjords and surrounding waterways. Bacteria from melting glaciers in Alaska sharing plasmids with other microorganisms they encounter in our oceans may be a mechanism for the introduction of pathogens with novel abilities to cause disease into our environment. Despite the practice of using subtherapeutic levels of antibiotics in animal feed for decades all over the world, the mechanism by which this practice promotes growth in animals is still not fully understood. Recent research indicates subtherapeutic doses of antibiotics alter the animal's gut microbiome in several ways that cause the animal to become obese. Killing off specific components of the gut microbiome, including lactobacillus and alobaculum, and the loss of diversity, are likely important drivers of the alterations in metabolism. Research has suggested low doses of antibiotics decrease the nutrient needs of the animal's gut microbiome overall by reducing the population of microorganisms while preserving those needed for nutrient absorption during digestion. Other factors that may work to enhance growth in the setting of subtherapeutic antibiotics include alterations of the animal's mitochondrial function, leading to changes in the immune system and metabolism, and inhibiting subclinical infections that otherwise would have diverted the animal's energy towards fighting them. We know that humans are exposed to chemically active antibiotics, both from environmental exposures to antibiotic residue in animal waste, and more directly from consuming animal products contaminated with antibiotics. But more research is needed to determine whether there may be a causal link between the human obesity epidemic and the widespread use of antibiotics in our food system. Antibiotic-resistant infections cause a significant amount of morbidity and mortality in the United States and globally every year. There's been evidence that antibiotic use in food animals leads to antibiotic-resistant bacteria, and that these same resistant bacteria in food animals are transmitted to humans for a long time. The World Health Organization made both these conclusions in 1997 based on an analysis of existing evidence at that time. More recently, there's been many specific examples strengthening the evidence of the link between the development of antibiotic-resistant bacteria in food animals and human health. For example, the rise of fluoroquinolone resistance observed in animal salmonella isolates has been associated with a subsequent increase in human infections through the contamination of eggs. There's also been a strong correlation demonstrated between the use of fluoroquinolones in food-producing animals and the emergence of resistance to ciprofloxacin in both animals and humans. There's an ongoing 2025 outbreak of salmonella tied to eggs, but a comprehensive review of antimicrobial resistant salmonella outbreaks tied to poultry in the US was completed in 2022, some of which is summarized here. In 2021, there were 1,204 confirmed human infections with antimicrobial resistant salmonella tied to poultry, resulting in at least 2 deaths. Salmonella cases and hospitalizations tied to poultry in the US have continued to increase on average every year, and multi-drug resistance among human salmonella isolates has remained consistent at 9.3%. Because of increasing antimicrobial resistance in salmonella, few antibiotics are available to treat patients with severe infections. In 2019, the U.S. reported extensively drug-resistant XDR Salmonella typhi, resistant to all antibiotic classes except macrolides and carbapenems. In 2016, member states of the World Health Organization requested a priority list of antibiotic-resistant bacteria in addition to tuberculosis, which had already been identified as a priority previously, to support research and development of effective drugs. The researchers tasked with developing the list selected 20 drug-resistant bacterial species to prioritize based on a complex analysis of many factors, including prevalence of resistance in the species, transmissibility, and treatability. MRSA and VRE made the list for high-priority gram-positive bacteria, while the list responsible for community-acquired infections was topped by clarithromycin-resistant Heliobacter pylori, fluoroquinolone-resistant Campylobacter species, gonorrhea, and Salmonella typhi. International organizations, including the World Organization for Animal Health and the World Health Organization, have developed global standards for appropriate use, reporting templates, and a global database to track sales and usage by antibiotic class and animal species. These frameworks are important because they enable cross-country comparisons by standardizing data and shed light on where gaps in data remain, notably in low-resource settings. Efforts by the World Organization for Animal Health to improve surveillance and antimicrobial stewardship have proven effective to improve biosecurity, animal vaccination programs, husbandry improvements, and decreased use of critically important antimicrobials. Some countries, such as Sweden, now require a veterinarian's prescription for any antibiotic to be used on animals. California also started requiring a veterinarian's prescription for antibiotics to be used in animals when it passed Senate Bill 27, which also banned antibiotics for disease prevention in livestock. Researchers used this policy change as an opportunity for a real-world experiment to evaluate the human health effects of decreasing antibiotic use in food animals. The researchers found that antimicrobial resistance rates decreased among E. coli populations contaminating California chicken products in the years that followed implementation of Senate Bill 27. They also found a corresponding decrease in resistance to extended-spectrum cephalosporins in E. coli from the urine specimens of patients with urinary tract infections in California after passage of the bill. The graphs show the difference between predicted and observed proportion of urinary E. coli isolates resistant to extended-spectrum cephalosporins by month from 2013 to 2020, before and after implementation of California's Senate Bill 27. On the left is specimens from all women, and on the right is specimens from all men. The vertical dotted line indicates when the bill was implemented. International organizations have also advocated for data transparency, which would likely increase accountability for countries and agricultural producers that don't use antibiotics responsibly. In addition to policy change and policy enforcement, monitoring of antimicrobial use, environmental monitoring, and preventive measures, additional research is also needed to decrease the reliance on antimicrobials in food production and to further our understanding of the health consequences of widespread antimicrobial use and subsequent resistance. Research to develop new antimicrobial agents is also needed, but we cannot innovate our way out of this problem. Following the golden age of antibiotics from the 1940s to the 1960s, not many new antibiotics have been developed, and there's been even fewer medical advances in antifungals. This is in large part because of the low return on investment for pharmaceutical companies. Most antimicrobials are only taken for a short period of time, so from a business perspective, investing in an antibiotic that people usually take for about 5 days doesn't make much sense when you could instead be investing in insulin that people with diabetes have to take for the rest of their lives, or the new popular weight loss medications, which people have to keep taking to avoid regaining the weight they lost. Drugs required to treat chronic diseases create lifelong customers. Another factor that makes developing new antimicrobials less profitable is, let's say there's a new drug that works well against both fungal infections and multi-drug resistant bacterial infections. We wouldn't start prescribing that all the time. We would save it as a last resort because it would be such an important drug. We would only prescribe it to someone with an infection resistant to all existing drugs. So, a low volume of sales would result from this new life-saving discovery. Antimicrobial resistance itself also puts drug company profits at risk. If they spend millions on research and development and eventually bring a new antibiotic to market, but then we overuse it and resistance develops, then we'll stop using it because it's no longer reliably effective, so the company is left with a worthless product. Controlling antimicrobial resistance is not a simple process. Bacteria don't care about national borders, and their trade of resistance plasmids and their other tricks cannot be regulated by human governments. This problem calls for a global One Health approach to implement regulations across countries, along with global monitoring and enforcement. Action is required across sectors, including human medicine, veterinary medicine, and agriculture. A reduction in the usage of antibiotics in animal husbandry is crucial. While significant progress has been made in raising awareness and promoting responsible use of antimicrobials globally, the lack of new antibiotic drugs under development and the continued high use of antimicrobials in food animals in developing countries underscore the imperative need for continuous efforts to combat this important health issue.